Hello. Today we're going to continue on our quest of network inference for cellular signaling, and in particular we'll try to infer signaling networks from temporal omics data. I'm going to use phosphoproteomics as an example, but similar concepts will apply to transcriptomics or metabolomics or other kind of omics data. Now this task of network inference from temporal data comes in a wide range of varieties. So let's look at the number of scenarios, and extreme ones are shown here on the left and on the right. And what this represents is our time axis is going from the top to the bottom, and every node represents a protein. The shade of blue represents the level of phosphorylation of this protein. So the very left is an extreme scenario because all the information that we're using here are phosphoproteomics data. We're not using any additional information to build our network. So what you see is this protein is getting phosphorylated first, then increases over time. These ones here are coming in second, and then at the end, in the last time step, this one here is showing phosphorylation. So we may decide that our network maybe should look something like this. So this protein is phosphorylating here based on the temporal order and here. And then on the third time step, we may say, okay, this protein here is phosphorylating here. But as you already see, there is already some ambiguity because it might as well be this protein here doing the phosphorylation, right? So I selected the lower one because this one has a higher level, arguing that this one is maybe more likely to phosphorylate here. But at the same time, you could also make an argument that it may also be this one, or it may be both of them at the same time. So as you already see, if you don't have any other information than your phosphoproteomics, then this is a very difficult task, and you will need very good data to infer a network, especially very good data in terms of temporal resolution. Now, if we look at the very right, this one is an example where essentially we already have our underlying network given. So we already know the edges in the network, and the task that for us remains to be done is to put values on these edges. So for example, in our case, this would be kinase rate constants maybe. And in this network, we might say, okay, the phosphorylation here seems to be relatively fast. This one here is a bit slower, but also still relatively fast. And this one here is maybe the slowest connection. This is based on the idea that these two time steps maybe are similar at least. And then what you might say is maybe putting values that reflect the speed, so something like this here. However, this scenario is something that doesn't arise frequently in the real world. So typically, we don't know our underlying networks a priori. In many cases, we do know some of the connections, so we do know certain kinase substrate relationships, for example. In most cases, we probably don't know all of them, and even more importantly, we don't know which connections we should be sure about should be absent from our network. So what we're going to do today is look at this middle scenario here. And the middle scenario is essentially one where we have our phosphoproteomics data, so in the order in which the shades of blue change, but we also have this underlying network. However, in this case, we don't want to put complete trust into this underlying network. So in many cases in the literature and in the cases that we talk about today, these would be protein-protein interaction data. And it's not just that we maybe say, okay, there may be false positives in this interaction data or false negatives, but there's also the additional issue of that a protein-protein interaction doesn't necessarily mean an interaction that's relevant for the signaling. So our task is also to tease apart which of these interactions are actually relevant for the signaling process and which ones are essentially just bystanders that don't inform us on the flow of signals through the network. Nonetheless, we use this interaction to guide how we think the information is flowing. And in the network shown, you might say, okay, based on these temporal changes and on the edges that we have available here, we would maybe infer that this edge is active. And then we might say, okay, this one we probably would put on as well. At the last time step, we would say, okay, it's probably this connection which we activate. Additionally, we might have said, okay, there could have also been a signal flow that goes through this node here, even so this node is not changing. So the one that I depicted is probably the more likely one. So this one is the paper that we want to talk about today and that we want to try to understand together today by Budak 2015. And this one is about inferring a signaling network based on just the data that I was talking about before, phosphoproteomics data with underlying protein-protein interactions. And they use a combination of two approaches. One is called Eichsteiner-Forest, and the other one is called integer linear programming. And based on these approaches, they are trying to infer this network from those temporal phosphoproteomics data. Let's just start thinking about which kind of solutions we might come up with to infer signaling networks from temporal data, from data where we have measurements on several components of our potential future network. And then invite you to just pause the video here and actually think yourself what you might come up with. So let's say we have data like this. X-axis is the time axis. The y-axis in some way represents a change in phosphorylation, and each of these nodes would be a protein. And then we see, okay, at this point, there is no change compared to the initial time point. And then here, once the points move up here, that would represent this protein now has changed by a large degree in its phosphorylation. Okay, so a simple thing that you may have come up with is simply saying, okay, well, apparently this yellow protein changed its phosphorylation first. So let's just give this protein a 1. And then we see next it's the green one, and then the red one and the blue one. So we would just say, okay, we can give this one a 2, we can give this as 3, which is not visible here, 3, and this one will get the 4. And therefore, because this is the temporal order by which they changed in their phosphorylation, we'll just connect them up like this. And this is the way how we inferred our signaling network. So very easy, very straightforward. But now, of course, we said, well, what we wanted to use is not just this omics information, but we wanted to use some underlying protein-protein interaction. So for this, let's look at a slightly more complex scenario, even still far from the complexity of a real-world scenario, of course. And what you see here is that we have these number 2 nodes. And so the question becomes, okay, through which of them should our information actually be flowing, or through both of them maybe? And then what you also see is these dashed edges here, which represent the connections from the protein-protein interaction network. And in this case, you can see very clearly that our protein-protein interaction data suggest that the flow of information should be via this lower path here, whereas the node number 2 up here doesn't have any connections. And with this, we're ready to introduce the ILP, the integer linear programming approach, which is described in this paper here. So I should say what I'm describing may not be exactly what's in those papers that I'm mentioning, but at least the idea is the same. And what we have is our signaling nodes and the underlying protein-protein interaction network, which is represented by these dashed edges. And we have numbers on these nodes which describe the direction in which they should be visited. So starting from a source node with number 1 to a sink node with the highest number. And our task now is to introduce a connection, always in increasing numbers from the source to the sink, with introducing the least possible number of changes to the network. So that means every time we introduce an edge that's not given by our underlying network, we incur a cost. So for example, we might say here, okay, we choose this additional edge, then we use this edge here, which is present, and then we introduce another additional edge, which would give us a cost of 2. It depends then on our design choice of what we're trying to do. If we include a punishment for having edges connected for which we do not observe a change, or if we're okay with this and we're just ignoring it. Or an alternative solution could be that we're saying, okay, we're taking this path here, and then we introduce this edge here and introduce this edge here. So again, we have introduced 2 edges that were not part of our protein-protein interaction data. So again, we have incurred a cost of 2. Now there are, as I said, different design choices on how you want to implement this. In the cases so far, we said, okay, we only want to arrive from the source node to the sink node. We want to find one path how the data is transmitted. You might say, okay, no, my priority is actually to connect all the nodes to make sense of every node. And in this case, you might say, okay, I actually want to have this connection, but I also want to have a connection from here to here so that I have an input for this number 2 node. And I also want to have this connection so that I also have an output coming from this node. Again, this depends on your design choices. So much for integer linear programming. But now in the paper that I introduced earlier on, we had these two approaches. We had the integer linear programming and what was called the Steiner forest approach. So why do we need something in addition to this integer linear programming? And for this, let's look again at a slightly more complex scenario. And what you're seeing here is that we actually don't have any direct unbroken connections from 1 to 4, but we have two paths in which we have these white nodes. So the white nodes represent proteins where we don't observe any change in phosphorylation. And of course, if we do integer linear programming, we are allowed to introduce new connections. So we can say we introduce this connection here, or we introduce this connection here at a cost. So this is not necessarily a problem, but nonetheless, we might wonder, okay, maybe it would still better to use the connection via these nodes, even though we don't observe a change in phosphorylation. So this may be false negatives where we fail to observe a change, or where we were just unable to completely observe this protein or the phosphorylation on this protein. And I'm not going to talk about how we're going to consider this yet, but this will have to do with how much value we give to nodes. So for example, if we say we trust very much in the change in phosphorylation in this node, this is a very high change, versus this one is very little, then at least we know that we would potentially prioritize this pass over this pass. And then the second thing is we would consider how much do we believe in those connections here? Like, are these interactions that we trust in a lot, or are these connections that we have little trust in? So when we want to take this approach of prioritizing certain connections, asking for certain nodes that are not observed with changes in phosphorylation, but still where we want to consider, should they actually be part of our network, then we can do this once we have assembled the network as it is here already. So once we have put these numbers on. But if we want to enumerate all the different possibilities, then based on how valuable we consider nodes, based on how much we trust in connections, then this may be computationally very expensive to go through all of these different possibilities once we are at our final network. Maybe the smarter approach would be to consider this at every time step. And apart from the computational cost, there may also be a reason for doing this simply based on saying, okay, at every single time step, the data should make sense. So for example, at the time step number 3, we should ask, okay, why do we observe a change in phosphorylation here? Is it possible that this information is maybe flowing along this route? Should we maybe include this node? And there is in particular an argument for this also because our choices of the source and of the sink nodes are somewhat arbitrary. So the source node was simply the first time step at which we generated data. Number 4 is the last time step at which we generated data. At number 4, we say we want to find the optimal solution, but the choice of number 4 is somewhat arbitrary. We could just simply have measured one time step more or one time step less. So there is no reason of not saying, okay, already at the time step number 3, we also want to have a best possible solution. So here's a summary of the approach that we're going to take. So the integer linear programming means we're going to introduce numbers on our nodes, and these numbers are going to come from at which time point does a node become part of a subgraph that we consider as reasonable, that makes sense in terms of the cost benefit. And then we simply try to connect from source to sink with the least number of alterations in this network according to the underlying protein-protein interaction network and according to following the numbers that we have on the nodes. And then the Steiner forest approach, which we're going to talk about next, is the approach where at each time step we're trying to define what is the most sensible network, what is the graph that gives us the best cost benefit. So let's talk about the Steiner forest now. And again, the idea behind the Steiner forest is to at each time point find the optimal graph, so the graph that gives a good balance between the benefits that we get and the costs that we have to pay. We'll first start talking about Steiner trees rather than Steiner forests. And what you see here would be each node again represents a protein, and the edges represent an underlying protein-protein interaction network. The filled circles are our required nodes, so these would be proteins that change significantly in their phosphorylation between, for example, before and after stimulation. So we want these proteins to form part of our signaling network. The numbers that you see on the edges are edge costs. So this actually represents a lack of confidence in these edges. The higher the number, the less we trust that there indeed is an interaction between those proteins. If there is no edge at all between two nodes, that would mean we have zero confidence, we have a complete lack of trust that there is any connection between those proteins. But also the higher numbers, we rather want to avoid using these connections for our signaling, because we don't really trust very much in that there is indeed this connection between those two proteins. You can think about this essentially as a traveling salesman problem. So the traveling salesman has to visit these required cities, and he wants to do so by having to pay the least possible amount for using the streets between the cities. So far, we're not talking about directionality, so it's not about visiting these cities in a given order, but it's only about, okay, include all of the required nodes. And if we want to look for a naive solution, then we could, for example, just eyeball this and say, well, if we want to connect all of the required nodes, we go along this path here, and we go along this path here, which, however, may not be the cheapest solution. So if we think about it a little bit more, we may see an alternative solution, and we may say, okay, let's use these edges here instead. And this one means we have only incurred a cost of 6 now. So this is a better solution. So what you see is that we have now gone through this node here, which is not a required node, but there is also nothing that forbids us to go through this node. And actually, this is a main point of the Steiner approach, that you can also include nodes that are not required nodes. So this may, for example, be a false negative in our phosphoproteomics data. We didn't observe a change of phosphorylation on this protein. Maybe we didn't observe the protein at all. And nonetheless, with this approach, we are allowed to go through this protein and make it part of our signaling network. And the reason for this is that even though we don't have any data from phosphoproteomics or any encouraging data that this should be part of our signaling network, we have good confidence from our protein-protein interaction, which makes up for this and which suggests, okay, we should use this node instead. These nodes that are not required nodes, but nonetheless become part of our derived tree, these are called the Steiner nodes. So as in other lectures, we're not going to talk about algorithmic implementations here. So we're not going to talk about how do we find those cheapest trees that lead to a connection of all our required nodes. The how obviously is an important question, because in this case we would have 7 edges. So if you imagine we enumerate for all of the edges, the edge either being present or absent, then we would have 2 to the 7 solutions, so 128 for a simple tree like this. And then we would have to ask, okay, does this solution give us a connection of all of the required nodes? What is the cost? And figure out the cheapest one. You can see that this obviously is not very efficient, especially once you go to larger trees. So the how question is important, but on the other hand side, like in lectures like this, people often start talking about the how, and then they get so lost in the algorithmic implementations that they lose the focus on the why and lose the bigger picture view. And we want to keep this focus on the bigger picture here. So let's go back to our Steiner tree, and let me point out again that the requirement was that we want a fully connected subgraph of all of our required nodes. So all of our required nodes have to form this tree. And I'm claiming that this is a problem and that this is something that we would like to relax as a requirement. So as an example, let's look at this tree here. And if you look at it for a while, you see that there are two subtrees on the left and on the right. If you want to connect up within them, you can find relatively cheap connections. So if you look at the connections in the left one, we can see we can build a signaling tree like this, which gives us a cost of 9. On the right hand side, we can use these edges here, which give us a cost of 11. And you see that we get those connections with relatively low cost compared to when we have to connect between them, because we only have one solution how we can connect between them, which is going along these edges, which gives us a cost of 21. So how this may arise biologically is, for example, if the connection that I drew here is not even the right connection, if the cell takes a different connection, but we don't know about it because of a false negative in our protein-protein interaction data. So it could be that there actually is a protein here which connects, let's say, these two guys and which connects them in a relatively cheap way, but we don't know about it because we didn't observe this protein in our protein-protein interaction. It's a false negative. We can even think about a situation where actually a stimulation affects two parts of the cellular signaling network independently. So it may not even be the case that there has to be a connection between all of the signaling that we observe. Even so, I said before that we really want our signaling to make sense at every time point. So essentially that we do want to end up with a completely connected tree at each time point. It's good if we relax this requirement a bit and allow for these possibilities of parts being stimulated independently, or especially of the possibility of false negatives, that we say, okay, we just don't know in which way two observed changes in a signaling network are related. So what is the solution? How can we introduce this possibility of not requiring a complete connection in our tree? And for this, we're now going to make the transition from Steiner trees to Steiner forests. And if you want to learn more about this, you can look up this paper here. So let's go back to our example tree. And what we're doing now is we're introducing a new node, a helper node. And then we're going to randomly connect this node to two of the existing nodes in the tree. So let's say we randomly connect to those two nodes here. Then we're going to ask, is the tree that we can build now with this new node cheaper than if we didn't have this node? And what's important here, of course, is the parameter of what is the edge cost for these edges that we introduced. This is an important parameter, and our algorithm will have to find a way to figure out the best choice for this parameter so that the new solutions are most sensible given the biology. Let's say in our case, this parameter is determined to be 3. So we're putting an edge cost of 3 on both of these new edges. And then we can ask, do we get a cheaper solution if we include this node? And in this case, it's obvious it's not the case, because now we have a connection that costs us 6, replacing a previous connection that was only 3. Just another word on this new parameter, on this edge cost for the helper node. So this obviously reflects our confidence in how much do we believe that our underlying data will allow us to build a signaling network. So if we believe that actually the underlying data are not very good, we have a lot of missing values and we can't build a single tree out of them, then we would put this parameter very low, so that in the end we end up having a lot of small disconnected trees. And on the other hand side, if we have a very high trust that we have very complete data and we should be able to build one single network, then we set this parameter very high, so we end up with essentially a single tree. So to estimate this parameter, we somehow need to get an indication on how reliable are our data, how much do we believe that we can build a tree from what we have. And in the best case, what we would use for this is a signaling network that we know is true, and then we would train the estimation of this parameter by using the signaling network, using the same underlying protein-protein interaction data, and asking, okay, where will this parameter end up, so that it reflects our ability of building the true tree out of the data that we're using. Okay, let's go back to our example tree. and keep trying to avoid this expensive connection of which we said we don't have complete trust in that it's the true connection that's taken biologically. So we'll keep trying further placements of our helper node and use random connections to required nodes. So let's say in this case, we end up choosing these nodes as the connected nodes, and let's say our parameter that defines the edge cost on the helper node edges is still 3. Then we would end up with these new introduced edges of a cost of 6. And then if we want to connect the further remaining nodes here, we actually wouldn't use this edge here, which gives us a cost of 5, but instead we would keep using this connection. So altogether we have 3 times 3 plus 2 plus 1, so we have a cost of 12. If you remember, the original solution was this sub 3 here on the right part, which had given us a cost of 11. So this is still not a cheaper solution or a better solution by introducing the helper node. So we can still keep going and try further solutions. And at some point we will end up placing our node so that it randomly connects up these nodes, and that indeed now we replace this very expensive connection of 21 with, let's say, our solution that still costs us 6. And so we have now found the best possible new tree, having introduced this helper node that replaces the very expensive old tree. So what I previously referred to as subtrees, so this part here on the left, this part here on the right, we would now just call trees. So trees as being defined as parts of the entity that are only connected via the helper node, and the complete construct now we would call a forest. From this point on, we will no longer consider the helper nodes. This was only a helper, this was only an aid of finding these trees and finding the best part of subdividing our original tree into a forest of multiple trees. At this point, you can now imagine that you keep going with this, so you can try to again place helper nodes and ask, can we also split up those trees into further trees to potentially replace connections that are so expensive that we don't believe they should be part of our final solution. So now we have come relatively far. So we have a situation where we have this network and we have certain proteins that we want to include in our signaling. We said we can overcome the problem of having false negatives in our phosphoproteomics data by including these Steiner nodes, so we can go through nodes even so they are not, they don't show a significant change in phosphorylation, so we can avoid problems due to false negatives. Again, if they're connected up well in our underlying protein-protein interaction network. And we have introduced this idea of the Steiner forest, that we say, okay, we don't need to use only the existing connections, but we can introduce additional ways in which we essentially artificially connect the up sub parts of our graph. And in this way, we can avoid problems due to false negatives in our protein-protein interaction. What is still a bit unsatisfactory at this point is that we say, okay, we have these strictly required nodes, so they significantly change in phosphorylation, so they have to be part of our signaling network. And then other nodes where they don't reach the significance threshold, and therefore we say they are not required. And real-world data are not really this black and white. If we say something is significant or not, then this usually comes from, okay, we place an arbitrary significance threshold, but we lose all of the information of, okay, a certain protein may be close to the threshold, other ones maybe just above the threshold, other ones may be very clearly above the threshold. And what they did in this paper that we were talking about today is saying, okay, we represent the fold changes or the log2 fold changes that we observe in the phosphorylation, and we don't actually place a cutoff anywhere. And this is what brings us now to this idea of price collecting Steiner forest. So again, we have our edge costs based on the protein-protein interaction network, but now we also have node prices, and these node prices are represented by the shade of the blue here. And again, these node prices in this case are selected as the log2 of the fold change in phosphorylation of these proteins. And now we don't have a situation anymore where we say certain nodes are strictly required nodes, other nodes are non-required nodes, but we have, okay, if you manage to visit a certain node with a certain price, then you get this price. If you don't include it, then you're not getting this price. And then you're making a trade-off between visiting certain nodes and gaining the price and paying the cost for getting to the corresponding node. So we have to introduce an additional parameter here, which gives us kind of a scaling between this node price and the edge cost, because this is a comparison of apples to pears. This is not something that we can directly compare on the same scale. How much we should weigh this price against this cost. So we need some kind of parameter that tells us what's an appropriate scaling, and this is something that should be determined by our algorithm, and in the best case should be determined on a ground truth on a network that we know, where we know how it's connected, and hopefully that reflects our situation so that we can infer these underlying parameters. Similar as determining the parameter that we had before for splitting Steiner forest into trees, where we say, okay, how much should this cost for the edges on the helper node be? So at this point, we have arrived at a pretty good understanding of the approaches in the Budak 2015 paper that we wanted to explore, of the Steiner forests and the interlinear programming. And now, before we move on and try to discuss the advantages and the disadvantages of this approach, let's maybe take even one more step back and let's ask, well, why do I make such a big deal of this inference of networks from temporal omics data? And I do really think this is a very important approach, especially in the context of phosphoproteomics and proteomics. So let's see how this approach relates to other possible ways in which we may do network inference. So one context in which we as a scientific community have done a particularly good job in inferring networks is in the context of transcription factor networks. So here each node would represent a transcription factor which binds to these promoter elements shown in the same color and then regulates the transcription of this next transcription factor here. And the way how we typically infer these networks is by chip, by chromatin immunoprecipitation, where we cross-link the transcription factor to its DNA elements. Then we share the DNA into smaller pieces. We reverse the crosslink and sequence those pieces, and thereby directly know which pieces of DNA are bound by the transcription factor. We can then further align these sequences, derive consensus motifs, and once we know a consensus motif for a transcription factor, we can ask, does this sequence occur elsewhere in the genome? And we can specifically test, does the transcription factor bind there as well? Did we miss that in the chip assay? So this mechanic or mechanistic information works very well for inferring these transcription networks. Unfortunately, in the case of phosphoproteomics, the same kind of mechanistic information is much harder to come by. Yes, you can do co-IP assays where you ask, for example, okay, which other proteins does a kinase bind to? However, if a kinase binds to another protein, this doesn't mean that this other protein is a substrate of this kinase. Whereas for transcription factors, if they bind to a given DNA element, then it's quite likely that they also actually regulate the gene associated with this DNA element, with this promoter. In terms of something analogous to these consensus motifs, yes, there are for kinases also consensus motifs around the phosphorylation site, but often these are not as predictive as for transcription factors. So often there are additional or replacing other factors that determine how a kinase binds its substrates that go beyond simply a linear sequence that we could infer, but once we know a number of substrates for the kinase. Another approach that's frequently taken to infer networks is by manipulating the network somewhere and then measuring outputs at other points in the network. So, for example, you could overexpress or delete a protein. So let's say we're deleting this protein here, and then we're measuring the output, let's say phosphorylation at other places in the network, and we see that now these proteins here are no longer getting phosphorylated. And if you then do this for multiple proteins in this way, you can build up your network. So you might also say, okay, now let's delete this protein here. And then you should observe no change in the phosphorylation of these proteins, but this protein should be phosphorylated less. If you measure in a quantitative manner, you might say, okay, maybe now we're only getting a quarter of the phosphorylation or so. A problem with this approach is that by making these manipulations, you might introduce changes into the network that are unintended and that go beyond the local changes that you would expect. So the network on which you're doing measurements might actually no longer represent the network that you're studying. So to explain this, let's again look at this scenario where we deleted this protein here. But now let's assume that the cells that don't receive phosphorylation here actually are not viable. So this is an important pathway for the cell. So the only cells that will then survive and on which you then can actually do measurements are the ones that somehow circumvent this situation of having this deletion. And even if they're not able to directly revert the deletion, which could, for example, be done if you only introduce a mutation rather than a deletion. But what could, for example, be happening is that they in some way strengthen this connection here. So this connection, which before was only a very tiny connection and which maybe you only find under very specific conditions where the cells are, have differentiated in a particular way or something. They may now use this connection that can potentially exist and through genetic or epigenetic changes make larger changes into the network so that they just retain their viability, and therefore the network that you're actually doing your measurements on does not reflect the network anymore that you are intending to study. So just to recap, so these direct mechanistic studies that we have seen for CHIP can be very helpful, very informative in inferring networks. However, they don't apply to every kind of network scenario. So for example, for phosphoproteomics networks, they are much less accessible to use these techniques. And then we have seen that this approach of manipulating the network can also be very informative, but here we have to keep in mind these unintended consequences, these adaptations to manipulations that may lead us to inferring a network that actually does not represent the wild type situation before the manipulation. A second bigger picture question that we might ask about what we talked about today is what is the usefulness of protein-protein interaction data. So we started off saying there are these different scenarios from which you can start for building a network, inferring a network. The one where you have no underlying information at all, we said that's hard if you don't have really good data. The other extreme where we said we essentially already know the network, a situation rarely arises in reality, and then this intermediary approach that we took today of saying we have a network. However, we don't want to completely trust this underlying network. And in the examples that we use today, what is out in the literature mainly being used are protein-protein and interaction data. So how useful is this? And I would say many biochemists would argue that data on just immunoprecipitations, co-immunoprecipitations, are not very good for informing on kinase-substrate relationships, simply because this is a fast interaction. This is considered a kiss-and-run interaction. And if you try to pull these proteins down together, then usually this interaction is too transient that you will be able to detect most of these interactions. Of course, there are other approaches. There are approaches such as yeast to hybrid that are potentially more qualified to give you these transient interactions. Something that I also want to say, the information on direct kinase-substrate relationships is not the only thing that we can get. That's by no means the only thing that we can get from this protein-protein interaction information. So let's look at an example. So here A phosphorylates B, then the phosphorylated B binds C, and now C, since it's bound to phosphorylated B, can phosphorylate B. So what you see here is that information from the protein-protein interaction, namely that B is binding C, can be helpful for us in inferring this network, so in particular inferring this edge here. even so the interaction between B and C is not a kinase substrate interaction. So this is what I meant by the protein-protein interaction network can be helpful beyond telling us directly about kinase substrate interactions. Now, what you do notice, however, is that in this example, this still is a dynamically changing interaction. And so we, on the one hand side, might ask, okay, can we maybe obtain data on dynamically changing interactions that may be more helpful for us than the static interactions? And secondly, this brings up the question, okay, how easy or how hard will it be in detecting this interaction? Will we arrive at a similar situation as for kinase substrates where it's hard to detect transient interactions, and maybe we again need techniques like yeast to hybrid to identify them in the first place? And then, of course, there are other data which are not exactly protein-protein interactions, but can be used in a similar way to protein-protein interactions. For example, co-expression data or genetic interaction data, which may also help us in building these networks. Overall, I would say the jury is still largely out on how helpful are both these other kind of data and the protein interaction data for these kind of approaches. So let's talk a bit about the advantages and disadvantages of this combined Steiner Forest IOP approach. And let's get started with the advantages. And one of the big advantages of this approach is that it handles false negatives quite well. And this is both false negatives in the phosphoproteomics and in the underlying protein-protein interaction network. So false negatives in the phosphoproteomics are handled via these Steiner nodes. So even so we may not observe a change in phosphorylation on a protein, we may not observe any phosphorylation on the protein at all, we can still have it as part of our network if we have a high confidence in terms of its connection in the protein-protein interaction network. It handles false negatives in the protein-protein interaction network well, and this is based on the Steiner forests rather than Steiner trees. So as we talked about, the Steiner forests allow us that we have places in which our network is not connected. So rather than saying, okay, we force a connection on the network that we don't actually believe in, because there may be a false negative, this step allows us to actually reflect this confidence that we may not have the complete knowledge and there may be something missing. Another advantage is that phosphoproteomics changes are reflected on a continuous scale. So we said we're doing price collecting Steiner forest, meaning there are prices on the nodes which reflect the level of the changes or the confidence level that we have in the existence of these phosphoproteomic changes. And on the other hand side, also our confidence in the protein-protein interaction network is reflected on a continuous scale. This is done via the edge costs. So the less confidence we have, the higher an edge cost is going to be. And reflecting something on a continuous scale obviously is a good thing because it gives you more information than if you just go binary and say something is present or absent. Every time you make this step from going from continuous information to binary information, you also lose information. So now I just said that one of the big advantages of this approach is that we're looking at our data in a continuous way, but there is still a way in which there is also discreteness, and this discreteness is one of the problems or one of the disadvantages of this approach. And this discreteness comes in terms of that we are considering the time points as discrete. So meaning when we build our Steiner forests and then decide at which point should a protein be included, as a number in the interlinear programming, then we essentially do this by looking at the current time point and not considering any other time points. So even so, of course, phosphoproteomics data, other kinds of temporal omics data, we will always get data that are discrete time points. There are different ways in how much we actually consider the data over time rather than just looking at individual time points in the approach in which we analyze them. So to make this a little bit clearer, let's look at an example for this. So the way this algorithm is thinking is we have our data. Let's say this is the time axis, and this one is something that represents something like a delta in phosphorylation. And then we would assume that our data looks something like this. So we have one protein that changes in phosphorylation here, and we have another one that changes in phosphorylation here, and the third one here. And then obviously it makes sense that we say, okay, the first protein came in first, so we say this one probably phosphorylate or can phosphorylate the second one, and the second one can phosphorylate the third one. So this is kind of how we imagine this data look like, and there this approach makes a lot of sense. But now, who is to say that our data really will look like this? Let's assume we have another protein in there which looks more something like this. And now, even if we say we don't put a significant threshold on how we select these data, we are still having this discreteness of saying at a certain time point, this new protein will be part of our Steiner forests and therefore will receive the given number then to be part of our integer linear programming. And so let's say this occurs around here. So around here, we say, okay, there is enough price on this node now that the algorithm will detect that it should now be part of our Steiner forest. Since we have at this point said this protein becomes part of our network, we might say, okay, the red protein was first and therefore may be responsible for phosphorylating the blue. But as you can see from the data, this doesn't really make sense because the blue one has already started rising earlier than the red one, so it's very unlikely that the red one would be the cause for the rise in phosphorylation in the blue one. It could be the other way around. It could be something like that the blue one is causing the change in phosphorylation in the red one because this one started earlier, but the other way around is unlikely. And therefore you see that if we only consider these discrete time points, this is going to be, we're going to ignore some information that may actually be helpful for us and lead us to a better interpretation of our data. Related to this is that the approach is always only backward looking. So when we ask about building our Steiner forest, we look at the given time, we look at what happened previously to decide if a phosphorylation chain should now become a part of our network, but it's not forward looking. And in this case, we might actually say, okay, if we were forward looking as well, then we would see, okay, there is a trend in the change of phosphorylation of this blue protein. And if we were to consider also this future data, then we might be able to convince ourselves that, okay, this change in phosphorylation actually occurs very early on. Whereas if we ignore any kind of future data, then we'll be much more limited in at which point we can actually detect that there's a change in phosphorylation going on. So this is what I mean, that this is a problem that we're integrating our data in the Steiner forests at discrete time points. Another big disadvantage in the approach that we talked about is that we have to make choices at several points and that we may not be able to actually make good informed choices. So what I'm talking about is on the one hand side, the parameter choices in the price collecting Steiner tree. So you remember when we talked about the Steiner forest, we had this parameter deciding on how much should our forest be split into trees, which we said should somehow reflect how much do we believe that there are false negatives in our data. On the other hand side, we also had this parameter that weighs between the node prices and the edge costs. In the integer linear programming, this was not so much about parameter choices, but rather about design choices of how we want to set up the integer linear programming. I already alluded to these design choices in the integer linear programming when I introduced the concept, but just to recap quickly, one thing that we discussed was, do we only want to find one path in which the information is flowing from our source to our sink, like this, for example, or do we want to say, no, we want to make sense of changes in all of the nodes, in which all of the nodes should have an in edge and potentially also all of the nodes should have an out edge. Another important choice is, what about these nodes where we don't observe any change in phosphorylation? Do we want to penalize those, or do we want to say, no, it's just fine that they remain in our final network? And another actually very important issue is, do we say we only want connections in increments of 1, so only going from 1 to 2, only going from 2 to 3, or do we want to allow bigger jumps and say, as long as something changes earlier, it can cause any change in a later time point, even if it's not in an increment of 1. So would we, for example, also allow introducing a connection from 1 directly to 3? Or in a very extreme case, would we even allow a connection directly from our source node to our sink node? So for example, if you think about the protein-protein interaction network, that's very large, and then we observe phosphorylation changes in all parts of this network, but our source node and our sink node are actually located very closely together. Maybe they even have a direct protein-protein interaction edge. Does it really make sense to require the information to flow throughout all the network and then return back to the sink node? Or would it make sense in a case like this to say, okay, why not allow the information to flow directly from the source to the sink? And this also has to do with how closely spaced are your phosphoproteomics data. If they are extended over hours and we say something changes in the first minute, and then we claim that this causes a change in something after an hour, this maybe doesn't make so much sense. But if they are spaced over seconds, then it may very much make sense to say something that changes very early on, and then something changes at the latest time points, which is only after 10 seconds, then the change in the first time point could still directly explain the change in the last time point. And so again, you have to make all of these different choices in our approach. And of course, having to make choices, like having to fit parameters, that's a part of life if you want to do any kind of modeling, essentially. The question is, how do we do that? And usually the solution would be to take some ground truth, to take some real-world data, and then say, okay, based on this ground truth, we fit our parameters, we make our choices. In this context, that just seems very difficult, and I don't believe there are very good examples out there where people have done that, simply because we would need a network where we have a very high belief in that the network is correct, not just that the edges that we see are true, but also that the absence of edges, that, according to our network, that they are indeed absent. So this lack of ground truth makes this parameter estimation and finding these design choices very difficult. What was done in one of the papers, for example, for the parameters in the price collecting Steiner forest, was that they said they went through different parameter values and then they looked for robustness. So do we find places in our parameter space where if we change the parameters, then the outcome of the solution doesn't change that much, which is a good solution, but nonetheless falls short of having this actual ground truth, and based on the ground truth, making those choices. And of course, this ground truth is also a ground truth that should pretty much reflect the scenario where you're working on. So you don't need to just have a good signaling network, but you actually should have a signaling network that you trust in that it's true, but that also reflects the data that you're currently working on. And this probably is not a disadvantage for the particular approach that we talked about today, but this is a bigger problem with all of network inference, that we would just need a better ground truth, a network that we have a strong belief in that it's correct. So a final criticism of the approach is that it results in a single graph. So in the construction of our Steiner forests, we will say at a given time point, either a protein becomes or does not become part of the selected tree. In the interlinear programming, we say an edge either is or is not selected for what we consider the correct network. And this is opposed to probabilistic approaches, where you result in a network where you say, okay, a given node, a given edge has a certain probability of being correct, has a certain probability of being changing at a given time point. And maybe this is not so much a problem with the Steiner ILP approach than rather with the implementation that we talked about, because you could imagine an extension of this approach that makes it probabilistic. So why would we maybe prefer this probabilistic reflection of our results? So systems biologists like to approach problems in this circle between experiment and model, where they perform an experiment, and then based on this experiment, they are going to construct a model. And then based on the model, they again design new experiments with the intention that these experiments then give you further information to refine your model. So if we say we have a model that's an all or nothing model, so we either say, okay, this model is correct or this model is not correct, then once we design a new experiment, essentially we only have the choices of say, okay, we once again accept our given model based on the experiment, or we say it doesn't really fit the experiment, so we have to throw out the model completely and start with a fresh model. In contrast, if you have a probabilistic model, then the data from the new experiment might just mean, okay, you have to tweak parts of the model. You might have to change certain edges in your model rather than having to completely throw out your model if necessary. So in this sense, the probabilistic model is to be favored over the model where we say, okay, we either completely trust in it or we don't trust in it. So now if we want to use the approach of having the circle between experiment and model, having the options of refining our networks over subsequent experiments, we have to ask the deeper question of, is this even going to be possible? Especially, is this even going to be possible if we go to different scenarios? So for scenarios where we maybe stimulate the cell a bit differently, or maybe we use a different cell line at all. Or is this approach only going to be viable if we repeat the experiment under exactly the same experimental conditions? So let's look at this made-up scenario here, where we say A phosphorylates and activates B, and phosphorylate B then phosphorylates and activates C, and phosphorylated C activates D. And let's say this is a scenario that we inferred based on given data, and now we want to ask, is this going to be helpful to use this information, or is it maybe even hurtful to use this information in a different scenario where the cellular signaling network looks a bit differently? So this could be the new true scenario that we want to apply it to. So this could, for example, be a different cell line. And what we see here is that B is not expressed and therefore is not going to be part of this signaling. Or you can think about several different options in why this network may be looking differently. In this other cell, expression could be one thing. It could be that B, for example, is localized to a different part of the cell and therefore doesn't take part in this signaling. And what we believe as a true underlying scenario is now that C is getting phosphorylated via an alternative pathway. And again, the question then becomes, if we now try to incorporate this information from a different scenario, is it going to be helpful or is it going to be hurtful? And what we can see is that having this edge that we previously inferred from phosphorylated C to phosphorylated D can actually be helpful if we say, okay, there should be a higher probability of having this edge because we observed it before. Whereas for these other edges, this would actually be more hurtful if we're saying, okay, there should be edges from A to B and from B to C, and then in this case, they are actually not there. Now, if we say we essentially only make these edges part of our underlying network, so to a similar extent as we would say protein-protein interactions between these proteins, then that could be okay because we say, okay, there is a potential connection, but in the end, we don't use this potential connection because we don't see the phosphorylation of B. What could be even better is if we say, okay, we observed these connections previously and we make them conditional connections. So we say, for example, B phosphorylates C, but only conditional on that we're also observing a phosphorylation in B. And similar also for this one, which we then in the end do want to end up using, we can say C phosphorylates D, conditional on that C is getting phosphorylated. And then it also doesn't matter if C is getting phosphorylated in this way that we inferred previously or in a different way. And especially if we don't want to add this new data at the step of the underlying network, but actually if we want to add this in the step of interlinear programming, for example, if we want to say we go away from this idea of removing any edge from the network gives us a cost of 1, but maybe we want to say some edges should be particularly costly to remove, then it might make sense, okay, to have these edges that we inferred from a different scenario to make very costly to remove. But then it's even more important to have these additional edges and say, okay, if we do observe the phosphorylation of B, then this is an edge that we want to be very unlikely to be removed during the integer linear programming. So in one of the future lectures, we will actually talk exactly about this idea of building probabilistic final models rather than a single final model. And overall, you see there are many things to be discussed, many potential advantages and disadvantages, many ways in which you could do things slightly differently or even largely differently in applying this way of network inference with some underlying data. I would be excited if you use this space here to start this discussion or to continue this discussion, and we'll then keep going with different approaches in the next lecture.